So in the last video for this chapter, we're going to talk about how genetic background and environment might play a role in different types of gene expression. Penetrance and expressivity are often getting confused by students, but penetrance is the percentage of individuals that show at least some degree of expression of the mutant genotype in a population. And so what you're looking at here is, does a trait show up? What percentage of people that have this gene are going to have this trait? Expressivity is going to be the range of expression of that mutant phenotype. And so what you're looking at here is, how does the trait show up? And we often talk about them in conjunction with one another. Expressivity can also be the result of genetic background differences or environmental effects. So looking to see whether or not a trait shows up. For example, there is a trait involved with the number of fingers and toes that you have. And sometimes, if you only get one copy of this, if it's a dominant allele, it might not be expressed in a heterozygous individual. If you happen to have this trait, it's autosomal dominant. If it had 100% penetrance, you would expect 100% of affected individuals to have additional fingers and toes. And a single copy of this polydactyly allele is usually sufficient to cause this condition. However, not 100% of people who happen to carry this dominant allele express this trait. And so the penetrance is going to be less than 100%. A different example, they happen to be one that involves with cancers. Familial autonomous polyposis coli, FAP. We know that it is 100% penetrant. If you inherit this disease mutation, you will get the disease. And again, this is provided you live long enough into adulthood. There are more studies going on right now on different types of disorders. There are some that are known to be involved with breast cancer, like BRCA1, BRCA1, and 2. And we know that they're heavily involved with many female cancers, such as breast cancer and ovarian cancer. However, just because you have that gene does not mean that you're automatically guaranteed that you're going to get breast cancer or ovarian cancer. It's just that it is a highly likely chance, and we know that women who do happen to carry these will often get prophylactic mastectomies or ovarectomies just to remove the organs before their cancer has a chance to develop. Expressivity, again, is to look at how does this disease or trait show up. So one example of this would be hereditary non-polyposis colorectal cancer. We know that it's about 80% penetrant, so maybe 80% of the people who have this are going to have this particular disorder. However, it also has variable expressivity. So looking at the penetrance first, if you inherit this disease mutation, there's an 80% chance that you will get the disease, again, provided you live long enough into adulthood. The variable expressivity comes into where it can show up in other systems. So maybe not just the colorectal region, but uterine cancer, cancer of the stomach, small intestine, pancreas, ovary, kidney, ureter. I mean, any of these different issues. And so the expressivity of this particular gene can act in many different ways. Genes can also be affected by their position or their physical location of the gene. This can influence its expression, and what we're talking about here is its position relative to other genetic material. So if a gene happens to be very close to a region with a lot of heterochromatin, we'll talk more about that later, or if it's heavily methylated, or if there's some other issues in that particular area, you might not get a very high gene expression from that particular region. And so that may cause you to have a lower gene expression that causes physical position effect. Sometimes you can look at things like effects of temperature on expression. So there's an evening primrose. If you have red flowers, it'll produce red flowers at 23 degrees Celsius. If it's a little bit cooler temperatures, it'll produce white flowers, 18 degrees Celsius. Things like Siamese cats, Himalayan rabbits, are going to have darker fur on cooler areas of their body, their tail, their feet, their ears. And what we know, if you think back to Biology 111, we talked about enzymes. Enzymes can lose their function, their catalytic function, at higher temperatures. And so at these higher temperatures, you may just see these different effects because the enzymes aren't able to function as fully. In the lab, as we talked about before, you may get these temperature-sensitive mutations. In this case, the mutant allele might only be expressed phenotypically at whatever temperature you have it working at. Usually it's a lower temperature. There are genes that are called heat shock genes. They're only going to be turned on if literally it's been shocked with heat. So you can get these higher temperatures. Your body will be able to respond to that. Here's an example of the rabbit and of the cat. You can see, again, that the cooler areas are going to have the darker fur. The warmer areas are going to have a lighter colored fur. Conditional mutants, we talked about the temperature-sensitive mutants. They're often considered to be conditional mutants. They're affected by temperature. And so these are also very useful in studying mutations that affect essential processes. So if you want to study something that affects an organism early in development, and you have a mutation in that gene, normally that organism would not survive so that you could study them. However, if you make it a temperature-sensitive mutation such that it is functional at lower temperatures, but not functional at the higher temperatures, you can allow it to grow at the lower temperatures, and then when you want to study it, you can increase the temperatures. There are many examples of nutrition affecting expression of genes. And many of these mutations prevent synthesis of nutrient molecules. So when we're talking about them in terms of microorganisms, like working with bacteria or yeast or fungus, we call them auxotrophs. And this phenotype can either be expressed or not expressed depending on the diet. Some examples, many of them from humans. So fetal ketonuria is you lose an enzyme that is used to metabolize phenylalanine. Phenylalanine is going to be one of the amino acids. And you can have very severe problems unless you restrict phenylalanine within the diet. If you do that, then since you don't have the enzyme to metabolize it, you're totally fine. Galactosemia can be very bad. Lactose intolerance can be just unpleasant. Again, it can be controlled by the diet. Individuals can avoid these things in their diet, and they can be phenotypically pretty normal. These nutritional mutations also often used to study things like genetics, especially in things like bacteria. What you can do is you can supplement the missing nutrient in the medium. And in fact, this has been used quite extensively historically in genetics. Beadle and Tatum, we'll talk more in chapter 14, but they use this sort of technique to determine the one gene creating one enzyme hypothesis. Onset, a time of onset of genetic expression can also affect things. So if you look at something like Tay-Sachs disease, this is autosomal recessive. It causes some problems with lipid metabolism. The baby may be born normal and may be completely normal for a few months. However, these do cause severe problems, and the child will usually die by about age 3. Lesch-Nyhan syndrome is an X-linked recessive disorder, involved enzyme defect, purine salvage defect. These individuals are normal for about 6 months, and then as they age, this becomes more and more of a problem. Duchenne muscular dystrophy is another X-linked recessive disorder. Children will often get diagnosed at about 3 to 5 years old. Even with advances in modern medicine, we still have issues, and it can often be fatal by the time individuals reach their early 20s. Huntington disease is an autosomal dominant disorder. It affects the frontal lobes of the cerebral cortex, but its onset is pretty late relatively. The onset for this disease is around between ages 30 and 50, with a mean age of onset at about 38. So you can see all of these individuals may present as completely normal at birth, but they may have problems either shortly thereafter or within a few years or even longer in their lifespan. Genomic imprinting and gene silencing. We'll talk more about epigenetics when we get to the section talking about gene regulation. But overall, what you should know is that it's a process that's going to involve modifying the DNA. It does not alter the genetic sequence. However, what it can do is it can alter expression of those genes that you do have. These marks are often made in utero. It can be established in the germline. They maintain throughout all the different somatic cells or normal cellular cells of this particular organism. And we know this happens a lot, especially in utero. There have been cases where you study individuals who maybe had poor nutrition during the pregnancy, and you can see the outcome in those individuals. It not only affects their offspring directly that were in utero, but it can also affect their grandchildren, because oftentimes the precursors for these germline cells are created while that individual was still in utero. And so nutrition, especially during pregnancy and things like that, incredibly, incredibly important. This whole inheritance process, however, is going to be independent of our classical Mendelian genetics that we've been talking about. And like I said, we'll talk about this more when we talk about gene regulation, because really what you're talking about is inheritance at the inheritance is not changed, but the expression of those genes does become changed. Conclusions for this week: please make sure that you read Chapter 4 in your book. Please make sure that you've gone through all of the lecture notes. Please make sure that you're up to date on all of the quizzes, all of the Mastering Genetic homework. Week 3 quiz will be available shortly. Please also make sure that you're making comments on the discussion forums. It's a fairly large portion of your grade, and like I said before, you don't have to have a 5-page paper on any of them, but what I do want is just some sort of a response on each of those, or at least as many as possible. The other major thing that you should do this week is start thinking about what you want to do for your student project. Start thinking about project topics or ideas, and I would like to talk with each of you individually, and I can help you decide and help you narrow down, because I think choosing a topic, getting started, I think that's probably the most difficult part of this process. And really, you should have your topic chosen by about the end of the week so that you can start planning out your topic, start doing an outline. If you're planning on doing independent research, we need to talk as soon as possible about what you're planning on doing with that. And thanks, and I hope that you guys have a wonderful week. Stay warm out there.